We have to eat protein every day because our proteins get worn out and then leave our body. To replace them, we eat the proteins that make up other organisms, break them down, and reconstruct them into brand new proteins. Brings a whole new meaning to you are what you eat, doesn't it? Well, how do we do that though? It seems kind of weird to take a potato protein and turn it into hair or fingernails or a digestive enzyme. The trick is we need to assemble them into a string of amino acids and then shape them just right to do their job. And here's how that works. So the critical thing to know about proteins is it's the shape that determines their function. It's a pretty universal concept in anatomy and physiology, that relationship between form and function. So it holds true of whole organs or even tiny molecules. So in a similar way, a garlic press, it does its job because of the way that it's structured. Even a rope is made out of lots of little threads, and the way they're woven together makes it stronger. Even a pillow, it's structured a certain way. Would it work the same if the stuffing were on the outside? I don't think it would. And what's the difference between just a sheet of paper and a paper airplane? Folding, obviously. It's the folding that gives the proteins their function because it gives it the shape. So here's an illustration of how proteins are structured in kind of 4 different stages. And I gotta take a minute out to give a shout out to Mariana Ruiz Villarreal, otherwise known as Lady of Hats. She's provided free to anybody who wants to use them these beautiful illustrations. So Mariana, if you're watching, thank you for providing this beautiful artwork. Well, protein structure starts with just the sequence of amino acids, and there are 20 different amino acids, so we have a lot of different ways we can put them together, kind of like the Lego example. And so these all just get strung out in a line, and then they get folded into smaller structures, either these little spirals or helices, or these sheets, and that's mostly based on hydrogen bonding. Those then get folded further into more complex shapes. And so there's our 3-dimensional structure of a protein. Very often, proteins will work as complexes like this hemoglobin here, where you have 4 different proteins all bound together, and they're little cages for this heme group with an iron inside that actually carries oxygen around the body. So there are 20 different amino acids, like I said, and these are the monomers that make up the proteins, or polypeptides. And they come in a whole different variety, from the simplest here, glycine, with just a hydrogen R group, and you can see all the rest of them highlighted here. All of these in the yellow are non-polar. Notice they have mostly carbon and hydrogen, so no polar covalent bonds there. There are quite a few that are polar, and those can then do some hydrogen bonding with other molecules around them. There are some that actually have positive charges and some with negative charges on the R group. Yeah, hold on a minute. So what is an R group anyway? That's a good question, Sal. I should have led with that one. So an R group. Amino acids all have a really similar structure, kind of like Legos again. So each one has the same structure, so they all fit together, but they do have some variation, whether it's in color or the chemistry. So amino acids all have this same chemistry in common. They all look the same. And so this central carbon, I'm going to focus there. Carbons have 4 bonds, right? So whatever is coming off of this, that is the R group. I think of it kind of like the remainder. When you do a division problem, that leftover bit is the R group. And that could be something as simple as just a hydrogen atom, or it could be something really fairly elaborate. Sometimes you're asked to identify the R group in a situation. And rather than memorize 20 different R groups, What I would look for is this. It may not always be arranged this way, but off of this central carbon we always have an amino group. Sometimes it's NH2, sometimes it has 3 hydrogens. A carboxyl group, sometimes this hydrogen is here, sometimes it's not, depends on the diagram. And this other hydrogen. Anything other than those, and of course the central carbon, is the R group. And it's that that gives it its chemical flavor and determines how the protein is going to take shape. And so when they're assembled into this big chain, and the red line just represents basically a really simplified chain of amino acids with just a few R groups hanging off here. So here we got this one with a positive charge and this one with a negative charge. That is going to create this ionic bond that sort of closes and holds this loop in place. You even have these guys, these hydrophobic interactions. These are all nonpolar. They're trying to get away from water. Here we have some hydrogen bonding between different polar R groups. And here we have this interesting one, a disulfide bridge. And that requires this sulfur-containing amino acid, cysteine. And these are actually covalent bonds, so they're permanent. And this actually leads to the idea of a hair perm, or permanent, which basically you treat the hair with chemicals that break these disulfide bridges, and then you reform them in the curl that you want, and it stays there permanently. So if the shape determines what a protein does, losing its shape means it would lose its function, right? And that's called denaturing. So if we take a protein shaped the way it's supposed to and shake it apart, that would be denaturing. It's different than digestion. Digestion would be, back to the Legos, breaking apart these different amino acids. But denaturing is unfolding. It's more like taking that paper airplane and unfolding it so that it doesn't work anymore. So denaturing can be temporary if it's just a little unfolding, or it can be permanent if it's completely unfolded. And of course, that's going to determine whether or not that protein can be used again or not. If it's completely denatured, it just needs to be torn apart and rebuilt. There are a few things that can cause a protein to be denatured. High concentrations of salt. If you think about it, you're putting those ions in solution, and they're putting chemical pressures on all of these hydrogen bonds, ionic bonds, etc., that are holding these proteins in just the right shape. Changes in pH. Each protein is usually built for a specific pH. Most of them neutral, but some are built to work in acidic conditions, like the enzymes in your stomach. So if you change the pH dramatically, that's going to change the chemical environment and change the shape. So does temperature. But really, once you get to a certain point, cold doesn't affect the proteins much. We don't put things in the freezer to kill them or denature proteins. We cook them, right? When we cook our food, when we get it up to a high enough temperature, usually over about 60 degrees Celsius, which I think is about 140 degrees Fahrenheit, then they're cooked. They're denatured, and usually permanently so, which is part of why we do that and cook our food. But you can also cook your food in a really low pH. If you've ever had ceviche, you cook the shrimp using an acid from the lime juice. We can also heavily salt things like beef jerky, and that kind of does the same sort of thing, even without high temperatures. A couple examples of proteins getting denatured that you may have seen on a daily basis. If you ever get a blister, it's usually because there's a lot of friction there, maybe a lot of heat, or even a sunburn or chemical burn of some sort. What's happened is the proteins that hold your skin, that upper layer, onto the surface have gotten denatured, and they let go and your layers of skin separate. So there's one example. Hair is another example. Hair is all made of protein. So if you use a curling iron, it heats up all those proteins, and as they cool back down, all those hydrogen bonds form in a new way. And so that's more of a temporary sort of denaturing. What's that? What about bedhead? Would that be an example of hydrogen bonding too? Yeah, good point. Bedhead would be a great example. You fall asleep and your hair stays in one position. It allows some of those bonds to reform temporarily, and you wake up and your hair is sticking all over the place. What do you do then? Well, you put water on it, you take a shower, spray it down with water, and that water then creates a new chemical environment and restructures some of the proteins, and then when it dries, it stays in that position. There you go. Lots of interesting and important applications for proteins and their shape. So there are proteins, absolutely vital to all aspects of life, whether you're plant, fungus, animal, or bacteria. And so we need proteins on a daily basis to replace the ones that are worn out. They're pretty tough, but they can be ruined if they get denatured. Well, that's it for this time. Until next time, remember, life is good.